respect to I just got the announcement. With respect to uh, uh, collating all of the previous work that's been done on fishes in South Bay wetlands. And a lot of this first started with me and Ali Weber Stover as we were working with the Wetland Regional Monitoring Program to develop guidelines for monitoring fishes. And one of the first questions that we had was what has been done, what works well, and how did they do it? And there were lots of different individual projects around, and we created an inventory that was really useful for developing those guidelines, which are now published and hopefully will soon be implemented beginning next year. Um, but that also highlighted how poorly we've been able to archive and collate all the previous data associated with those previous independent studies. And so I had a really good meeting with Donna and with Dave about whether or not we could try to gather some of that original raw data into a database format so that we could inform future monitoring, inform future restoration, and inform a variety of other data needs with respect to endangered species and so forth, with a single repository that actually has some raw granular data that can be used to inform those or to support those needs. And so we developed a project about a year ago, and I've been working on it since then. And today I'm really excited to have an opportunity to share where I'm at with that project. It's still ongoing, it's not complete, but it will be completed by early next year. And the main point of the study was to basically document all the previous work that's been done on wetland fishes in South Bay wetlands associated with habitats as part of the South Bay Salt Pond Project, to get those data into a single repository and then to be able to provide a bunch of summaries, including maps, general data summaries that would be really informative to anyone who's trying to learn about what's been done down there and has any major questions about fish habitats in there. So without further ado, I'll just jump straight in. And I just wanted to give credit to the South Bay Salt Pond Project for funding this data archival and collection effort. Okay, so I'm going to introduce my lab quickly. So we do a lot of work. Don already mentioned it with a lot of different entities, a bunch of different departments at UC Davis, federal and state agencies, and then a lot of local work as well, and in particular with the South Bay Salt Pond Program, San Francisco Estuary Institute, SFEP, the San Jose Santa Clara Regional Wastewater Facility, Stillwater Sciences, Valley Water, all the usual suspects. Many of you are probably on this call. And so this talk just focuses on this one aspect of our research, which is the local research. And most of the data I'll be sharing that comes from my lab was funded by all these entities on the right, and they've been wonderful to work with. And again, I didn't collect all the data. A lot of this has been collected by myself and also by my predecessors, especially Jim Hobbs. You'll see his name pop up a lot. And then I have a fairly large lab that also does a lot of the data collection for the ongoing and current research that we do. My lab specifically focuses, as Donna mentioned, on surveys and monitoring, which I'll talk about today, but also otolith analyses, both age growth and geochemical analysis, and then modeling those data to inform conservation. And so for surveys and monitoring, we use boat-based surveys, towing nets, deploying traps, and so forth to look at fishes. And then otoliths or ear stones, or we even use fin spines to do age and growth. And then we use ICP-MS machines to examine geochemical patterns in otoliths as well. We've done projects all across the world. We've published papers in Turkey. We're working with folks in China, as Donna mentioned. But again, a lot of our focus is here, and almost all of our fieldwork is in the San Francisco estuary. And so today, as I mentioned before, we're focusing on fish and fish habitats here in South Bay salt ponds, or South Bay wetlands. And it's all under the context of what can we learn about what happens to aquatic communities when we go from the left to the right as we store former salt ponds into functional salt marsh. So quick intro into fishes in wetland habitats. We know wetlands are declining across the world. This plot's just showing the global decline of natural wetlands across Earth. And we know that here in San Francisco Bay, the pattern's the same. We've lost 95% of our wetland habitats. Those wetland habitats, the vegetated component supports a lot of terrestrial or semi-aquatic organisms, but also the tides, as the tides come in and out, those terrestrial components are also interacting with aquatic components, and the aquatic communities are all highly impacted by this marsh habitat. The marshes obviously affect water quality. They also affect the availability of food, and also just the geomorphological structure of the marsh determines what species can be there. And in the aquatic habitats, the primary organisms that are there are invertebrates, such as either plankton or macroinvertebrates and fishes. And these fishes are using the marsh in very many different ways. This is just a conceptual model that we put into our monitoring guidelines for the WRMP. But there are intertidal habitats that are constantly getting wetted and then drying on tidal cycles. There are subtidal habitats that are staying constantly wetted. There are habitats that are in little tiny channels inside the marsh, bisecting more complicated vegetated plains. And then there are larger sloughs that are typically always wetted and are kind of semi-open water habitats that bisect larger marsh complexes. And then just outside of those marshes, you also have large open water habitats like shallow mud flats that are intertidal and also shoals that are more subtidal. And all of those are being affected by what's going on in the marsh. So anytime there's a restoration action, it's not just affecting what's going on at that location, but there's also lots of downstream effects. And all those downstream effects, whether it's increased sedimentation, reduced sedimentation, whether it's increased production, whether it's changes in water quality, it's affecting all these different habitats, both intertidal, subtidal, within the marsh and adjacent to the marsh. And there are fishes and invertebrates in all those habitats. And the only way to understand what's going on in those aquatic communities is to monitor them. Um, Here we're focusing in San Francisco estuary. I don't need to introduce this group to this system, so I'm going to kind of skip through this quickly. But mostly I wanted to point out that we kind of dissect this into the upper estuary and the lower estuary. And there's marshes in all of these different locations. In the upper estuary, we have Sassoon Bay and all of its marsh habitats. In the what we call lower, but basically just west of there, we have the San Pablo Bay habitats, and it has quite a bit of marshland. And then in South Bay, we have quite a bit of marshland as well. And there's a lot of information from individual projects in these habitats, but there really hasn't been a lot of good integrated monitoring in these habitats. And integrated monitoring is really valuable because that's the only way in which we can detect change, either due to climate change or due to restoration or due to other types of oceanographic processes. You name it, the only way to have a good, to be able to detect change is to have really good long-term monitoring. And that's exemplified in the State of the Estuary report. This is just an example of the most recent one in 2019, and we're currently working on the one for 2025. But that report is only functional or only feasible because we have really good monitoring data, for example, from the Sassoon Marsh study, from IEP surveys, and in particular in the lower estuary, the CDFW Bay study, otter trawl, midwater trawl data set, which has been going on for, well, since the 80s. And so in terms of marsh habitats, the only long-term, all these dots are just individual stations that have been monitored long-term by IEP. And the only places that we have in brackish marsh habitats that have been monitored long-term are in Sassoon, here highlighted, kind of in the upper right. But in San Pablo Bay and North Bay marshes, we really don't have very many dots, and because there just hasn't been long-term monitoring in those habitats. And that's problematic because that means that even though we have really good reviews, like the Fish Restoration Program and its effects on tidal wetland restoration, the effects of tidal restoration on fish, the USGS analysis and paper Physics to Fish, understanding factors that create and sustain native fish habitat in the San Francisco estuary. Those are really good, powerful papers written by really intelligent people with really amazing data sets. But because the data are limited to the upper estuary, they really focus only on the upper estuary. And the upper estuary is really different than the lower estuary hydrologically and biologically. And so that means that we have a somewhat biased understanding of how marshes function because we focus so much on the upper estuary and we have so little data on the lower estuary. And as I mentioned before, we can see changes in vegetation pretty easily. We can see changes in bird abundances. And if we look closely, we can even say changes in mice abundance and presence from land, but we don't know what's in the water unless we actually monitor what's in the water. And so a lot has been done, but again, many of these projects have been short-term projects, either 1 or 2 years, that have been directly tied to individual restoration projects or other individual programmatic needs of various entities across the estuary. And so they're disconnected. They use different methods. They have different standards of reporting. And it's very difficult to glean an integrated understanding of fish and fish habitats across South Bay. But what we wanted to do is bring that all together and do the best that we could to provide a best available science perspective on fish and fish habitats in South Bay wetlands. And so we've done that. And what I'm going to do for the next part of this talk is share the overall framework for how we're approaching this review, the studies that we've highlighted, And then I'll go through and just provide some examples because we don't have time to look at every single study. But I think the examples really highlight how diverse the different types of studies, how diverse the different types of studies and approaches that have occurred in South Bay are, and also how difficult it is to integrate them because they're also unique and different. And they also are so super valuable because they're diverse. They give us lots of different perspectives in terms of what works, what doesn't work, and what we should do in the future. And I hope to pull all that together and give you at least my perspective currently. And I hope that perspective will be fairly well supported. Okay, so this again focuses on South Bay fish studies. So I'm not including anything from other regions of the estuary, even though we have started compiling studies from the upper estuary and from the northern part, San Pablo Bay wetlands as well. But for this, we're just thinking about really Ravenswood, Alviso, Eden Landing, and maybe a little bit of Bear. Um, I also wanted to highlight before I start that there have already been some general reviews with respect to the South Bay Salt Pond Program. These are synthesis reports from phase one, phase two, and also the Mercury studies published from 2018 to 2022. You all are probably familiar with these, and a lot of the fish data is in there, but it's a pretty high level overview of the fish data. And again, the raw data, the granular data, aren't made available in these reports. And so that's one of the big things that we wanted to do was to basically take the data that inform these reports and make sure that it's granular and available to facilitate other types of analyses and other data needs. So historically, I've identified these 8 historical or semi-historical reports that talk about fishes in South Bay wetlands. So I have not obtained the Schmidt one. I just found it this week. So this is an ongoing project. But apparently there's a report written from 1912 to 1914 that talks about South Bay fishes, and I'm searching for it. If anybody knows where that is or has seen it, I would love to. I would love to get my hands on it and to include it in our review. But the first report that I was able to find was Carpelin, 1957. The study was in 1951. Carpelin was looking at fish communities in South Bay salt ponds specifically. Alpin, 1967, did a study not directly in the South Bay salt ponds, but in open water adjacent habitats from 1963 to 1966. And De Vlaming was a researcher from, I believe, UC Berkeley, who was examining longjaw mudsuckers in alveolo ponds. Specifically, he was looking at reproductive cycling. Um, and then Pearson used data from citizen science or an educational survey to examine fish communities, again, not directly in South Bay salt ponds, but in adjacent open water habitats from 73 to 82. And perhaps the most interesting data set that I found, one of the most detailed, has been an Ottertall data set from the South Bay Discharge Authority from 1983. And it encompassed some sampling in 81, but mostly 1982 to 1986. And it actually was sampling deep within Alviso Marsh along Coyote Creek and was examining, I believe, water quality parameters, but also took really good data on the fish communities that were there. And I'll share a little bit more about that in a minute. And then Mike Psyche and Meja from USGS, they published a couple of papers where they used gill nets to examine the immediate responses of fishes to restoration of the island ponds in Alviso. And so I'll mention those as well. So that's the group that I consider somewhat the historical literature for South Bay wetlands. And if you're aware of more, feel free to ping me. I'm happy to include additional data in this review. Since then, there's been a lot of additional work, and a lot of this has come out of my lab. Not all of it has been done by me. A lot of it was done by Dr. Hobbs, who started the lab quite a while ago. And I won't go through every single one of these studies, but you can see there's a lot of studies by Hobbs et al., and I will highlight a few of these. And these have all really informed how we think about wetland fishes in South Bay and how we're proposing to implement implement FFH monitoring for the WRMP across the entire estuary. And I also wanted to highlight that there are also some ongoing and future studies. So Tom Gass and Associates are working with Helix and Ducks Unlimited and Ravenswood and are collecting fisheries data. I don't have a lot of information on that yet, but I hope to reach out and chat with them soon. My lab is currently collecting data in the Calabaza San Tomas. As part of the Calabaza San Tomas project, we're working with Stillwater Sciences and Valley Water. And so we're collecting fisheries data inside and outside of ponds in the A8 complex and around that area in Alviso Marsh. My lab is collaborating with the Center for Watershed Sciences at UC Davis to conduct the first regional analysis of wetland aquatic communities. And so we'll be looking at fish and invertebrate communities in the Alviso Marsh, the Petaluma Sonoma Napa Marsh system, and the Sassoon Marsh system, and doing a comprehensive community assessment. So looking at differences in diversity, abundance, community structure, even from biomass between those major marsh systems across the estuary. And we're hoping and planning to begin monitoring as part of the WRP FFH, FFH's Fish and Fish Habitat Survey beginning early of next year. And it sounds like the funding just arrived, which we're really excited about. And I've already mentioned this, but I just also always want to highlight that there are already reviews out there that highlight some of this work. And it's always really helpful to always circle back. So this is basically the overview of my literature review. And these are all the different data sets that I am hoping and trying to incorporate into a single master database that'll be available to anybody who ever wants to work in South Bay wetlands. And I'm hoping to get those data as granular as possible. The stuff that we produced ourselves can be fairly granular, and it's a bit more of a challenge sometimes to look backwards through time and get those more granular data from the historical data sets. Even the ones from 2006 can be challenging. But I'm working on it, and we're getting there. This is what that master database looks like. Again, you probably can't see all the details, but we have codes and descriptions of all the different studies. We highlight the gear types, who sponsored it, who was the PI or author of the manuscript or paper, what was their affiliation, what were the study dates, what was the report date, do we have a report in our archive, do we have the data, the raw data, or at least some version of the data archived, and then what types of data are in that data archive. And then also we have report links to all of those. So again, this is one of the products that we hope to be able to provide, will provide to the South Bay Salt Pond Program and anybody who's working down there. We also have stuff not just on fishes, but we also have some papers that we found on zooplankton. I'm not including that currently in this review just because it's outside of the scope, but as I come across them, I'm archiving them because they can also be really valuable. And then also I'm documenting syntheses. This is a timeline of what some of those studies look like. So I mentioned there's a Schmidt paper from 1912. I'm hoping to find some of those descriptions of fishes. There's the Karpelin study in 1951, where again, he was using a dip net just to look at patterns in organism abundance in ponds across different pond types from low salinity to high salinity. Again, Alpin was doing a trawl survey, I believe, down in South Bay. I won't go into all these, but my main point, though, is that we have a spread of South Bay fish studies going all the way back to the mid or early to 1900s. And then we have a slew of them in the late 70s and early 80s. And then we have an explosion starting in 2006 for obvious reasons. The South Bay Salt Pond Program really started getting going. And that's where a lot of the new data has come from. Everything here in red has kind of limited data. It's mostly observational or doesn't have specific fish-related a fish-related focus, but has some fish-related data. Everything in black, we've already collated the data, or they have specific data on fish. Everything in blue is relatively new and is still in progress. So just to highlight what some of these studies look like, this is the Ecology paper published in 1957 by Karpelin. I think it's Lars Karpelin, if I recall correctly, from Stanford. And Lars was focusing on the Alviso salt ponds, what he labeled as 1 through 6, but it includes the A8 complex. He examined seasonal patterns and environmental conditions. So we're not only including fish data, we're also including environmental data in this database. And also examined just how salinity varied across those different ponds. And then he didn't do anything specifically quantitative for the biotic community, but again, used a dip net and did provide really nice descriptions of what types of fishes were there, really focusing on the fact that athorinids seemed to be really dominant across these different ponds and pretty tolerant of all the different environmental conditions. Another study going back to the 70s, this is by Victor de Vlaming, coming out of UC Berkeley, if I recall correctly. And what Victor Dr. de Vlaming was working on, again, was the patterns in longjaw mudsucker use in Alviso salt ponds, and in particular, the cycling of their reproductive patterns. So again, de Vlaming provides some really nice environmental data like temperature, which is shown here, and correlates those patterns, those seasonal patterns in temperature with a bunch of different reproductive metrics. upper left is showing testicular weight of male long-jaw mudsuckers, and on the right are seasonal patterns in the ovary, the weight of the ovaries in female long-jaw mudsuckers, again from 67 to around 1970, with the paper being published in 1972. So this is another great observation. If we're thinking about long-jaw mudsuckers as a sentinel species in our system, this is a really nice anchor point, and getting these data including them um, in our database is going to be really valuable. The Like as I mentioned before, one of the most exciting data sets that we came across was the South Bay Discharge Authority Otter Trawl Survey. This was from 1982 to 1986. Here you see a map of the Alviso ponds, and let's see if I can. Here we go. So these are the Alviso ponds, and where you ever see the stars, there's a star there, SJ4, SJ2. Um, those two major stations, there was long-term monitoring, long-term, during that time period in the 80s of the fish communities using otter trawl surveys. And what was really exciting about that is that they provided really good data on the fish communities in that slough network. And they use very similar methods as what we currently use. And so that provided an opportunity for us to compare changes in aquatic communities between the 1980s and present in these slough habitats, which otherwise we wouldn't be able to do with this data set. So here on the right, you can see Jim Hobbs pulled this figure together just looking at the total catch per unit effort from the 1980 to 1986. You have my pointer here, 1980 to 1986 period. And then our data from 2010 to, at this point, it was just up to 2016. So in general, CPUE seemed to be fairly similar. We had some much higher catches occasionally in our otter trawls. But generally speaking, it seems like we haven't seen a major shift in terms of total abundance, which was nice to see. In addition to that, we could also look at changes in the community composition. And I won't go into too much detail about it, but it is in this report produced by Hobbs back in 2017. And here you're just looking at the frequency of occurrence of a bunch of different species, from arrow gobies and Shokahazi gobies all the way to, on the right, shiner perch and starry flounder. And sometimes you see the blue and the red look pretty similar, like for northern anchovy, they seem pretty similar between 80s and current. Same is also true for Pacific staghorn sculpin. They seem pretty similar between 80s and current. But there are other cases where you see that there's a pretty big difference. So if we look over here on the far right for shiner perch, it seems like there were a lot more shiner perch historically. Also, there were a lot more starry flounder historically. There were a lot more longfin smelt historically, and there were a lot more striped bass historically. And just based on our understanding of what has happened in the Delta, it's not surprising that might also be happening in lower South Bay salt ponds. And so it seems like some of these patterns are a regional pattern, not regional, regional in terms of the entire SFE, but they're not sub-regional. They're exhibiting similar long-term declines in abundance of certain species. And there are some fishes that we just don't observe hardly at all anymore that historically were there, like dwarf perch. We do see some white sturgeon now, and a variety of others. And then also we did the same thing for invertebrates. And again, you can see like grass shrimp and oriental shrimp. Oriental shrimp is a non-native shrimp that's been around clearly since the 80s. But we also know Siberian shrimp appear to have been more of a recent invader. And actually, this only went to 2016. If we include our data to 2024, the Siberian shrimp population has exploded all over the South Bay salt ponds. So using these historical data sets are really informative for understanding changes that has occurred in the system. And this SBDA otter trawl survey is going to be a really valuable thing to have documented in our archive. And this is just a multivariate ordination examining variation in seasons and between the two different time points, but I won't go into details. I just included it in case somebody wanted to circle back and think about it carefully. Another example is the USGS gillnet and minnow trap study. So this was conducted by Mike Psyche and I believe Francine Meja. And they used experimental gillnets and minnow traps to examine fish communities inside and outside of restored salt ponds and the island pond system. And one of the questions was whether or not and how quickly do the restored ponds post-breach start supporting native fishes in particular, but any fishes and natural processes. And so they monitored inside and outside fairly shortly after the breaches, and provided some statistical analyses that basically showed that fishes pretty quickly started colonizing those unvegetated, shallow kind of mud flat habitats. And so those seem to be really important habitats. They were very rapidly colonized, and the populations were fairly similar. This is just a dendrogram examining distance based on community composition between the different stations that you can see in the map on the left here in Alviso Marsh. And the main point I want to point out is like Pond A19 is pretty similar to Coyote Creek Station 3 in the slough. Pond A21 is fairly similar to Coyote Creek 5-4 and even A20. And then, so basically, the restored ponds were clustering with the nearby slough habitats. So basically, the fishes that were in the sloughs were rapidly utilizing the new pond habitat, which was exciting to see. It's an indicator that that habitat is valuable, it is useful, and it's rapidly colonized by native species. Another example is the UC Davis leopard shark study. So moving away from some of those community analyses and now thinking more about kind of a sentinel species or single species approach. Jim Hobbs conducted a study where he was examining leopard shark abundance in some of the different salt ponds across the estuary using both set nets or gill nets. These are nets that are set and the fish swim into them. And then they can be collected and tagged and assessed for various health conditions like diet or weight and so forth. And they also use hook and line as well as another sampling technique. And so here's a map on the left from our actual database highlighting the different types of sampling gears that we use, angling, 3 different types of gill nets, and also even trammel nets, and the distribution of that sampling, again, across Eden Landing, Bear, Ravenswood, and Alviso Marsh. On the bottom right is just from our database highlighting the level of effort that occurred at each of those different regions for each of the different types of gear. So again, this is really valuable information if you want to understand the use of these wetland habitats, or just the presence or absence, or even just the timing of use of these habitats by large charismatic fish that we know are highly migratory and serve as a really important fisheries resource. This is a great archive or a great database to examine those patterns for this fish. And I just want to highlight that not only were leopard shark adults observed in these habitats, but it seems like these habitats are really important pupping grounds. You can see a little baby leopard shark there on the left held by Nick Buckmaster. Also, when a sentinel species is being examined, a lot of individual data can be collected. Again, length, weight, condition. In this case, they were examining diets. And so there's also diet data that we can include. And the diet data, in this case, you see a bunch of yellowfin gobies and 3-spine stickleback on the upper right image. And those individual types of data can also provide additional information that community data can't. So for example, if we just look at a single species, we might see that certain marshes are resulting in either higher contaminant loads, lower body condition, poor feeding, and so forth. And those are very different types of metrics than changes in community structure. And so it's just a different approach, and I'll highlight that again later. But this leopard shark study is a great example of that. And in addition to those types of data. So this is just catch per unit effort on the left of bat ray and leopard shark. And then also just highlighting, as I mentioned before, that we get all different size classes. So on the right is just the density plots of standard length on the x-axis and the density or relative abundance, or relative amounts of catch of leopard sharks in those size classes. And again, you can see there's a large peak at around under 250 millimeters, which are those pups, and then a bunch of peaks around 750, which are those adults. But in addition to the leopard shark data, this is what oftentimes doesn't get included. When you have a sentinel species project, there's other data being collected along with those sentinel species. And so in that data set, there's a ton of observations of other species in the gill nets and also caught by hook and line. And so we're going to include all of this. We've already included all of this into the database, and that'll be a really nice resource as well. Another short example, this was a fairly short study, but the intention of this study was to examine the outmigration of steelhead in the Guadalupe, kind of Coyote Creek, Alviso Marsh watershed. And the concern was that steelhead were getting entrained into some of the ponds. And so Jim Hobbs and colleagues electrofished steelhead up and down the watershed, inserted those. We assume they're steelhead. They were rainbow trout, which could migrate. I'll just call them rainbow trout for now. But they were all implanted with pit tags, and then those pit tags had transceivers placed on the opening to A8 to see if those steelhead could be detected moving in and out of the A8 complex. And there weren't very many detections. We didn't see a lot of steelhead coming out, but there were a couple detections And those couple of detections confirmed that steelhead certainly are able to and occasionally do move in and out of these pond structures. And I also just want to highlight that we don't catch a lot of steelhead in our monthly otter trawl surveys, but we have caught one fairly large adult steelhead migrating through the system, which was pretty exciting. So we know there's steelhead here, and we know they can move in and out of these systems. And the study just, even though there weren't a lot of detections, it highlighted that we're pretty confident that they are capable of moving in, and we can also identify around when might they be moving through here. In this case, for example, in February, March. I promise I won't go on forever, but I really want to just highlight some of these different studies and their approaches. This is another sentinel species study conducted by Jim Hobbs. This is the longjaw mudsucker. It's a large goby that lives intertidally on top of marsh plains. It lives in these pickleweed marshes that you can see here, and they tend to travel through these what we call rivulets, these small vegetated creeks that provide a lot of structure and a lot of foraging opportunity. At the low tide, these fish don't leave the marsh; they actually stay in the marsh plain and just burrow down into the mud. So they basically operate more like a salamander, and they're very tightly linked to vegetated marsh habitat, especially pickleweed dominated marsh habitat. And so they're a great sentinel species if you're thinking about just pickleweed marsh and variation in the condition of fish that come from different types of pickleweed marshes. So Hobbs and colleagues sampled these fishes with minnow traps. Here's a map from our database highlighting the different locations where those occurred. Again, the study occurred in Eden Landing, Ravenswood, Alvisa Marsh, and Bear. And they sampled a bunch of different habitats, intertidal habitats, managed ponds, tidal marshes, um, restored tidal ponds, and more open water habitats as well. On the right is just a heat map from our database highlighting the relative levels of effort. So all of this is already incorporated into our master database, which is great. And it's a super useful resource. And again, even though it's a sentinel species study focusing on longjaw mudsuckers, we catch lots of other things. And these are just catch data for all of the different species that were observed in that minnow trap study, which that haven't really been published just because they weren't the focus of the study. So this is now also has been incorporated into the database, and is going to again be a really valuable resource once it's fully available. And I just wanted to highlight from this study, there were two reports actually. The first one was actually using longjaw mudsuckers as a sentinel species. And then the same study was basically repeated, but instead also using staghorn sculpin as a sentinel species, which is instead of a goby, it's a sculpin with slightly different behavior. It doesn't stay in the ponds. It does go out into the sloughs at low tide. It moves back and forth, but also provides a really nice example of a sentinel species that could be used to examine variation in the health of a system based on variation in the health of the fish. UC Davis also has a mercury beach stain study that they conducted from 2014 to 2017. So this was part of the ongoing mercury study. One of the big concerns about salt pond restoration, as many of you know, is the mobilization of methyl mercury, which then can get into the food web, harming wildlife and harming people. And so mercury has been monitored in the system for a long time. In 2014, Jim Hobbs took over for Daryl Slaughton and continued to collect samples for mercury analysis. But in addition to that, also started doing quantitative surveys, which previously had not been done as part of the mercury study. So now, not only do we have mercury data, but now we also have quantitative community data that could be used to inform our understanding of patterns in aquatic communities across the estuary. And so he utilized a variety of different types of gears. I focus on beach seines, but he did try out fike nets and trapping as well for this study. Again, on the left is the map of the sampling stations from our database. On the right is a heat map indicating the sampling intensity by date and by station. And again, all of these data are going to be readily available. What was really nice about this study is it allowed us to examine patterns in the relative abundance or catch per unit effort of a bunch of different species across different types of habitat. So he was focusing on managed ponds, sloughs, and fully restored tidal ponds. And what we found, if I recall correctly, there were some differences, but overall, what Hobbs found is that very similar to what Mike Psyche found, is that the ponds are being utilized by a bunch of different fishes, both native and non-native, pretty rapidly and similarly to other habitats, whether they're managed ponds or sloughs. And so all these different habitats appear to be providing a lot of additional habitat for estuarine species that are living in the marsh, and likely overall increasing total productivity of the system. And again, lots of these small fishes serve as important prey items for larger fishes, fisheries targets, as well as for piscivorous birds and even other organisms like harbor seals. So it was a really nice example of how quantitative surveys can inform us about how aquatic communities vary across different types of marsh habitats and different types of management approaches. And on the bottom right is just the similar type of plot. Instead of abundance or catch, being, and effort, it's just looking at diversity, the total number of species. And again, they're relatively similar, though the muted ponds appeared to have higher diversity at times, likely because they also had a higher variation in environmental conditions because they had, well, there's a lot of swinging going on seasonally and diurnally. And then also those data, as I mentioned, not only were I'm interested in the community data, but also those studies also contributed to our understanding of mercury in the system. And I won't go into detail here, but it's all highlighted in this report that was produced by USGS. And then last, I'll emphasize just our South Bay otter trawls. So we've conducted otter trawls in South Bay since 2010, and these otter trawl surveys have really highlighted seasonal patterns, interannual patterns, and relative patterns of abundance for a bunch of different species, over 60 species of fish, 40 different species of invertebrates. And a couple of quick highlights. We've examined relative abundance inside and outside of the restored tidal ponds, A19 and A21 on the left. I'm highlighting that there's relatively fewer macroinvertebrates in those tidal ponds, but we see either more or the same number of fish inside the tidal ponds as we do in adjacent slough habitat. So again, those restored tidal ponds are really functioning well when it comes to fish. And we have three different pieces of evidence now. We have the Psyche paper, we have the mercury study paper, and we have the otter trawl paper. So the ponds are functioning really well for fish, and I think that's exciting, and those data all support that. We've also used those data to do regional comparisons, and we've shown that Alviso Marsh has up to 10 times higher density of fishes, that's on the far right, the bar on the far right, than all of the marshes that we've sampled in the upper estuary. And they have a different community. So on the right, far right, is just an MDS analysis of the differences in community structure between South Bay and North Bay habitats. And so again, this is another repository we're adding to the archive, or another data set that we're adding to the repository of the archive. And it provides really valuable information in terms of how do these marshes compare regionally, which is something that we've never been able to do before because we didn't have standardized monitoring. We've also highlighted the presence of longfin smelt in these habitats, which has been really important. Longfin smelt were just listed as endangered federally this past year. And so understanding their distribution is really important. Here I'm showing all three different life stages from pro-larva to juvenile to adult. And I'm showing dots. So there's purple dots, green dots, and even orange dots. And those are basically all showing individual catches. And the size of the dot is just the number of fish in the catch. What I want to highlight is that we're seeing larvae and juveniles and adults in a bunch of different habitats, but in particular, we're seeing lots of juveniles and adults in lower South Bay salt ponds and also in North Bay salt ponds. On the far right, if you look down in Alviso, you see these giant purple circles. So we've been seeing higher densities of longfin smelt in individual catches than is recorded anywhere else in the entire rest of the estuary. And that is really informing and modifying some of the policy. And it's been a huge resource for developing the longfin smelt broodstock program. So again, this data set is part of the South Bay Otter Trawl Survey. I call it SBOTS. And it's also going to be a really valuable resource for highlighting the distribution of this endangered species, but also all the other species that we monitor in the system. And we've also done intensive surveys as part of that. I won't go into too much detail about that, but that's also been included. And what those intensive surveys do is they provide us higher statistical confidence to do some more complicated habitat suitability modeling, and that's informing our understanding of the impacts of dissolved oxygen on fish communities in South Bay wetlands. And that's some work that we've done with San Francisco Estuary Institute. And these are just some of the occupancy model results that we've been providing as part of that program. Okay, so I said 30 minutes and here I am. But basically to wrap out, I just want to highlight one is that we've included a lot of different data in this archive. We found some really interesting historical data. We have a lot of data that has come out of my lab that we're including. And also there's some recent and ongoing and future studies I just want to highlight so folks are aware. So for example, Ravenswood is currently being monitored. for fish and fish habitat by Tom Gass and Associates. They're working with Ducks Unlimited and Helix. I don't have a whole lot of information there, but I'm hoping to coordinate with them and get that information. We're currently working with Stillwater Sciences and Valley Water to monitor the Alviso Marsh complex, specifically inside and outside of the AA complex. And that's to inform both, again, the mobilization of mercury, but also to get a better understanding of fish community and aquatic communities in those habitats using beach stain surveys. And that's our first real implementation of the WRMP monitoring guidelines. And so that's been really exciting to do some comparisons between otter trawl surveys and beach stain surveys using our new standardized guidelines. And so that'll be coming out soon. We just completed that this year, and we're currently writing it up. And then the South Bay Otter Trawl Survey continues. We just talked to the city of San Jose, and the city of San Jose is very excited to continue that long-term otter trawl survey in South Bay. So we're excited to continue working with the city of San Jose. And then in the future, as you all know, WRMP is cranking up, and we're hoping to start sampling more regionally across many of the different watershed units across the estuary beginning early of next year. And a lot of this increase in monitoring, the idea is to provide long-term standardized data that can then go into the state of the estuary report. And so we're providing a scoping report to the SFEP to highlight what kinds of data are available and what kinds of data we need in order to include a wetland state of our wetlands component to the state of the estuary report, which hasn't been done before. And in terms of lessons learned and forward thinking, as I mentioned before, fishes are using these ponds quite a bit, and we have several different studies have highlighted that the ponds are readily being colonized and occupied and utilized by a variety of different fishes, many of which are native fishes, and that's really exciting. In particular, one of those fishes is longfin smelt. Again, it's endangered, and it seems to really be loving these restored tidal and intertidal salt pond habitats, which is really exciting, was not previously known, and is really changing our understanding of how we should monitor this fish and how we should manage it. And then forward thinking, this is where I'll finish, is just a lot of this work went into informing how we developed the WRMP, specifically for fish and fish habitat monitoring. And so there were two major decisions we had to make. Are we doing the sentinel species where we look at one fish, or are we looking at aquatic communities? And we decided aquatic communities is better because we get so much more information when we think more broadly. And also it allows us to sample in different ways that maximizes the value of the data. And so that's why we're basically focusing on aquatic communities in that regard. Those special studies on sentinel species could be used. Methods, we had a lot of different methods to evaluate otter trawls, beach stains, minnow traps, fyke nets, and gill nets were most commonly used based on this review. And we settled really focusing on the otter trawls and the beach stains, though we also highlight that there could be a place for minnow nets and gill nets for special studies. Standardization is key, and it's really lacking. I hope it's pretty clear based on what I've just shown. And it's really needed to assess change, like I said, due to restoration activities or climate or other. And so these data repositories, though, even though we have data sets with a bunch of different methods and so forth, these repositories are super valuable so that we can inform future monitoring in terms of how do we do it, where should we do it, and then also for informing restoration and just knowing what species are where and when. And then last, the regional monitoring, integration, and quantitative analysis of wetland fish and fish habitat across the entire estuary is really needed. And what's really exciting is that it's planned and funded and it's starting next year. And I'll stop there, and I'm happy to field questions.
Thanks a bunch. That's really great and a really thorough review of all the existing data. I also, I'm just dropping it in the link in the chat right now. All the monthly surveys you can read about. Jim Irvin and now Jim Hobbs are writing about what they see in the South Bay every month. So those are really, really fun and informative little blogs every month. So I put that in too. So I will entertain questions from the audience. And you can either raise a hand, go ahead and unmute and just ask, or you can put your question in the chat.
Hey, Dr. Lewis, great talk. It's awesome work. Thank you. I put a question in the chat. California sea otter are losing ground. Their population's declining. North and southern limits of their range are declining. The bay used to be heavily populated historically by sea otter. Are there any plans to restore sea otter to the South Bay or any parts of the bay?
Yeah, that's a good question. I just had a chat with somebody who was thinking about that. And my understanding is that sea otters used to be pretty abundant. I don't think they were super abundant in this region, though I will acknowledge, let me start here. I am a fish person. So let me start there. But just having chatted with some folks that do study mammals, like I know we have a really nice and robust population of harbor seals that actually utilize the marsh habitat. We see them out feeding inside the salt ponds. We see them hunting fish on the marsh plain at high tide. It's pretty cool. And then we see them, of course, basking on the mud flats at low tide. So we know marine mammals can absolutely use these habitats. What I understood though is that the habitats generally aren't their optimal habitat. They'd probably be more abundant more towards Central Bay rather than in the South Bay habitats. Whether or not there's efforts to restore them right now, I don't think so. And so far, the efforts to translocate them have failed. They tend to keep moving back to where they came from. And so I think it's riddled. Ideally, it would be really amazing to have them back. But my understanding, just chatting with some folks that have been dealing with the otter um, challenge, is that it's unlikely that they're really going to put a lot of effort into it just because it's unlikely to be very successful. But again, that's coming from a fish person. I'm probably not the expert to really comment too much more on otters.
And then Christina Thoms put a resource into the
Yeah.
chat too for people. So um, let's see. There was a question, and now I lost my question about eDNA. And have you done eDNA surveys for specifically thinking about long fin smelt and other
Yeah,
species?
that's a great question. We have done eDNA surveys. In fact, we just published a paper this year for the first time where we tried using eDNA to detect longfin smelt specifically, and we were getting detections. We did this work with Liz Bowen. She was a lead author at USGS and other colleagues, and we did otter trawl surveys in South Bay and North Bay wetlands, and we highlighted that there are definitely longfin smelt there. We put our hands on them, and they also did the eDNA surveys, and they detected that they were there. They didn't always detect them. The probability of detection, if I recall correctly, was kind of 50-50, even though we knew they were there. But it really informed us on different methods that we can use to detect, and knowing what our detection probability is really valuable. And so that was the first step. It's published. The fact that you brought that up is a good point. I did not mention that in this talk, and I should include those data in my database. So thank you for that comment. The answer is yes, and it will be included. And the idea is that that's going to be expanded. That was a first look. I currently have a proposal being evaluated by the Delta Stewardship Council with a postdoc who wants to expand upon that work and do longfin smelt, steelhead, and sturgeon monitoring with eDNA in all of the wetland habitats that we're sampling for the WRMP. So that would be really exciting if that gets funded, but we'll wait and see. So there is definitely inertia, not inertia. There's definitely energy moving in that direction. Yeah.
So Brian Wines has a question. Are there standardized protocols for otter trawls and beach seines that other parties should use in sampling programs?
Yeah, that's a good question. So we did publish our guidelines as part of the WRMP um, SOP development effort. And so maybe Christina can post those in the chat as well. But also, if you reach out to me, I'm happy to share them with you. So yeah, there are standardized guidelines, and I will note that those guidelines basically follow what the Sassoon Marsh study has done for a very long time, since the 80s. And the reason we want to do that and follow the Sassoon Marsh framework is just because they have such a robust data set, and it's been a very successful and effective method for sampling large areas very rapidly. So there's a lot of trade-offs between the different methods that we use. And one of the nice things about otter trawls is that we can get to a lot of different places very fast and get a lot of data over a large geographic scale.
And then a lot of
Yeah.
the WRMP work, you'll be focused on sort of the effectiveness of using those methods, right?
That's right. Yeah. So we've already started implementing. We did side by side otter trawl and beach seines as part of our Calabaza San Tomas monitoring effort that we just finished this year, and we'll be doing a quick analysis of that. And we've planned to do some additional side by side comparisons of a couple different gears. But we already have recommended methods in the current guidelines, and I think those methods are pretty robust as they are. And if you want to follow up on any of those details, if anything's not missing or is confusing, please feel free to email me. I'm happy to go over that.
Great.
Definitely lots of do's and lots of don'ts that's worth chatting about before you start a program.
I
Yeah.
think I've got all the questions in the chat. If anyone else has questions, feel free to jump in. Oh, yeah. Are salmon being detected in any of the study areas from survey?
Yes, and I didn't put that in here just because it wasn't a formal study. But there were reports of salmon getting into I think it was pond A16. I might have it wrong. Quite a while ago. And Jim Hobbs did some gillnet sampling and was definitely catching Chinook salmon. We've seen dead Chinook salmon, and we know that Chinook salmon are moving up into some of these watersheds, I think, right now. They're being observed actually in some of the streams. So there are absolutely salmon moving through the system. They can get into the ponds, but they're extremely good at avoiding otter trawls. So an otter trawl is a terrible way to sample salmon. So we never see them in our otter trawls, but we know they're there. And that's one of the reasons why thinking carefully about what gear we're using is important because the gear that you use determines what you see. And so we know they're there, but they're not in the otter trawls. And so other methods like gill nets or monitoring the streams is a much better way to detect those salmon. But we know they're there. Yeah.
So let's see, how much, no, there was a question before that. Are you looking at fish abundance and water temperature?
Yeah, so one of the last modeling exercises that I showed, part of the intensive surveys, but also part of our long-term monitoring program, we always collect environmental data. That's also part of our monitoring guidelines is, we are fish people, but the only way to understand what's going on with the fish is to understand what's going on with the environment. So we are as much environmental monitoring, we're an environmental monitoring group as we are a fish monitoring group. So we monitor salinity, temperature, conductivity, pH, oxygen, and turbidity in a couple of different ways, both turbidity um, and Secchi depth. And so we understand water clarity, how warm it is, what the pH is, and how salty it is. And those are some of the major parameters that we think are really driving patterns in fish abundance. And that's always recorded. Our current guidelines, we actually take 4 measurements per survey, per individual tow, so that we know whether there's stratified water on the surface versus bottom, and whether or not there was a change between our beginning and our end of our monitoring effort. And so yes, the environmental data is absolutely critical. And I always emphasize with my team, in many ways, it's more critical than the fish data. The fish data can be a little bit messy, but the environmental data have to be really good, and our sondes have to be finely calibrated, and there has to be good calibration records. And that's all part of our guidelines for the WRMP. Yeah, that's a good question.
So Rick has a question about how much carbon is sequestered when a salt pond converts to a tidal marsh, and a good reference for that. I
That's
think
a, yeah,
we have some experts on
we
the
do.
call too who might be able to help with that. So
Yeah, I'll defer to another expert. We focus on the fish and the environmental conditions, but I'm not quite sure about the carbon sequestration. But I know there's a lot of folks working on that right now.
yeah.
Um,
We can, I'll just call her out here. Lisa Beers is on this call, and she might be able to, you might be able to reach out to her like Rick at SFEI, and we can get you a more complex answer and some references. So just a thank you from someone on the work and the presentation. So, and I can't see as many of the folks here, but is there anyone else with a question, I guess, or comment? Great work. Thank you, Levi.
sure.
Thanks so much for presenting today. And Levi traveled last night and he jumped up early this morning to give us this really great presentation. So if you have any questions for Levi or for us, feel free to reach out to me directly, or I can channel those questions to Levi too. So.
Well, thank you, Dave. Thank you, Donna, for inviting me. Thank you all for listening and for your amazing questions. Feel free to reach out directly. My email's there. My lab website is on the slide. Feel free to write that down or take a screen grab. And I'm happy to chat with anybody if you have additional questions or thoughts.
Thanks a bunch.
And definitely if you have any ideas of additional studies that I might have missed. I'm always taking new studies and recommendations. Thank you.
And
this was great, Levi. Thanks very much.
Yep.
Levi, was it the Schmidt paper that you wanted a reference for?
I want the actual document. Yeah.
I have that. I copied it out of the chat.
Oh, awesome.
It's downloadable, so I'll send you that information.
Thank you. That'd be great.
Yeah. Great. Thank you, everyone. Have a great day.
Bye, everyone. Thanks so
Bye.
much.
Thank you.
Hey, I should just notice that at peak we were at 68 attendees. When the talk ended, we were at 67, and that when we finally said goodbye, we were still up over 55. That's like a really, really good retention rate. We don't always get that. Seriously, that's like some people tune in and they like hear the first 20 minutes and bail. Sometimes it's lunchtime or whatever, but no, that was just really well presented, tight, compelling information. So
yeah,
I wanted to share that.
People
thanks,
like
Dave.
fish. It's great.
That too.
Okay, I'll shut us off.
Right.
Have a good day.
Bye, everyone.
All right, thanks, everybody. Have a good one.